we'll go from there. Today, we are going to have a presentation and a demonstration by Kapil Chowgule on new and improved tools for RNA-seq data analysis. Our presentation will take about 50 minutes with about 10 minutes for questions. Please, again, type your questions into the chat so that we have a record of them, as well as then Kapil can go through them at the end. And we'll have an opportunity also to post further questions if they occur to you afterwards on our Ask forum. Let's see what else we want to talk about this morning. Basically, panelists are the only ones allowed to speak, so you'll have to type in your questions, and there'll be time at the end, though, for discussion. So thank you very much, and Kapil, the floor is yours.
Yeah, thanks, Tina. Hello, everyone. My name is Kapil Chowgule. I'm a computational science developer here at Cold Spring Harbor Laboratory. I've been working on the Cyberse project for the past few years, especially working on the expression data analysis and integration of tools and workflows. So today's webinar will be focusing on some of the new and improved RNA-seq workflows that are available today for doing data analysis. So this, if you have used previously some of the Tuxedo workflows for doing RNA-seq, you might have encountered some difficulties in terms of the computational time it took to analyze like a large amount of data and stuff. So here we're trying to address some of those challenges with the newer tools, which have a better runtime in terms, and also they're more accurate compared to the Tuxedo ones. So what I will do in this webinar is basically give you an overview of those new workflows and try to give a benchmarking on how these ones have improved compared to the older suite of tools. So so before I begin, so if you have any questions, just please type in the chat box, as Tina mentioned, and we'll try to address them at the last 10 minutes. So so this is the outline that we'll be doing for the webinar. So I'll just give you an intro of what RNA-seq is. We'll go over the, we have the overview of the RNA-seq workflows, both the traditional ones and also the newer workflows. And then we'll spend the next 20 minutes or so on the demo. So I'll basically give you a brief demo of how you can run each of these applications. So I'll not run all of them, but I'll give you one or two applications that we can run. And also we'll demo another workflow, that is the Callisto one. And then we'll summarize some of the results that we do from the demo. All right. So RNA-seq over the years, so RNA-seq has been like the go-to analysis in terms of like finding differentially expressed genes or finding like transcript structures. So over the years, the cost of sequencing has drastically reduced. So as a result of which, RNA-seq has seen an exponential growth in terms of the number of publications and also data submitted to public repositories. And the reason also behind this has been the change in the sequencing technologies. There are improved methods or chemistries that have been developed where the sequencing process has not only improved the read length but also reduced the cost. So considering the data adults that you can see here, the analysis part, the software tools that have been available recently have also changed. So a lot of the tools are trying to address this big data bottleneck and trying to leverage a lot of the computational infrastructures to have an efficient method or an algorithm that can give you meaningful results in very quick time. So that's been how the RNA-seq has evolved over the years. So when you look at a typical RNA-seq experiment, so what you basically are interested in, you have certain samples of interest. where from which you isolate your RNA, and you generate the cDNAs for it, and then you put some linkers which go on a Illumina flow cell or some other sequencing platform. And what you get out of that is your sequencing reads. So it could be paired end or single end. And you align those reads against a reference. It could be a reference transcriptome or a reference genome. And that follows from there, you do your bioinformatic analysis. So overall, if you see, the most of the bottlenecks now have come down to the informatic analysis. So that's where some of the newer tools are helping out in terms of making it much more easy for even bench scientists to apply these tools and do the analysis. So if you look at some of the goals that an RNA-seq experiment tries to achieve, most probably it's relating to gene expression or differential expression. Some go with alternative expression analysis, or if you want to do a more qualitative analysis, you want to do a transcript annotation or allele-specific expression. And in case of cancer studies, if you want to find fusion genes, so you do fusion detection. So all of these analyses have specified experimental goals. So there is not one tool that satisfies all of these needs. So there's always a trade-off of which tool can be applied to which particular analysis. But in most cases, we'll be doing like gene expression and differential expression. So today's demo will be focusing more specifically on that aspect. And if you want to look at some of the strategies that you can imply with some of the research goals, you can there's a recommended reading for this one. So this review basically describes you, it gives you a snapshot of how things should be approached if you're looking specifically for, let's say, allele-specific expression and what tools that you need to use and so on and so forth. So I would recommend reading this one, which is pretty good survey on some of the data analysis methods. All right, so since we are focusing on differential expression, so I'll just give you a brief idea on how the workflow goes about. So basically you start with your inputs, that's your raw sequence data, and if you have already annotated reference sequences, so you can add in the reference annotations and the reference genomes. And then you need to do some pre-processing of the raw sequences, especially you want to remove some of the low quality bases. So there are already a lot of tools available like FastQC or a FastX toolkit or Trimomatic, which help you remove some of the low quality reads. So this becomes also essential in a way so that your alignments are more proper or are more meaningful. So once you have a filtered set of reads, you take those and align it with a spliceware aligner. So previously there was Top Hat, Star, and some others which did a pretty good job. And then you do transcript assembly. Basically, you take the alignment files and you do either a reference guided assembly or a de novo assembly if there isn't a reference available. And then once you have the assembled reads, you can do quantification. So there are different methods. You can do either count-based method or FBKM-based method. And then you want to do differential expression analysis, or you can do alternative expression followed by visualization using some of the R packages, which offer nice visualization features. So yeah, so that's the overall workflow on differential expression. So, but when you talk about differential expression too, there are two different ways you can do that. So one that I talked to you about is the alignment-based approach, where you basically, let's say if you have two conditions, you basically take the reads, you align them against the reference, and once you do the quantification, then you can compare the quantification using some statistical methods to do differential expression on it. And then the recent tools, some of the tools they skip this alignment step altogether, or they do something called a pseudo alignment or lightweight alignment, which essentially is they find the compatibility of a read mapping to a transcript or a genomic region. So there's a difference in how these two methods approach for doing quantification, and each has its own advantages. So in alignment free, it's very fast, and also in alignment based, sometimes you can get a lot of reads that are novel, which sometimes it's not possible with the pseudo alignment or alignment free approaches. So now when we saw some of the older tools, that is the traditional Tuxedo workflows, that's been around for a while and has been the standard or a go-to workflow for doing RNA-seq, and it has been adopted in a lot of projects in like the ENCODE, the modern code project. It was pretty easy to implement and work around doing getting differentially expressed genes or doing pairwise comparison. But the downside to this approach was it was computationally a bit slow. It demanded a lot of RAM if you're working on a big data, and some of the statistical significant results that you get for the differential expression seemed a bit more conservative. And if you had a complex experiment, it wouldn't have been possible to have like more than 2 group comparison with CuffDiff. So that was another challenge. So to overcome some of these challenges, so the newer RNA-seq workflow have addressed some of these needs, especially the newer Tuxedo workflow. It was developed from the same group that developed the TopHat workflow. So here in the new Tuxedo workflow, they have replaced TopHat with HiSat, cufflinks with StringTie, and they have replaced CuffDiff with BallGown. And so this particular workflow, the protocol runs almost on a similar line, but the underlying data, the algorithm for each of these tools and the data structure, the way they do the alignment or the assembly is markedly different from the older Tuxedo suite. So, and as a result of this improved algorithm, you can see the improvement in the speed and the accuracy of the results. On the other hand, then we have the other alignment-free tools, especially Callisto, Salmon. So these are some of the popular ones. So they also rely on the pseudo alignment or lightweight alignment in case of Salmon, which again has a different approach of how to do quantification. So these ones, so then Callisto also has a Sleuth, which is an R package that will do differential expression. So both these approaches have markedly improved the speed and the accuracy of the predictions. So in today's webinar, we'll be focusing on the HiSat workflow, and we'll be doing another demo with Callisto and Sleuth. So on Sleuth, I'll be showing you the visualization aspect on how you can visualize your results that you get from the Callisto analysis. So just to give an overview of how the HiSat workflow goes about, so you initially have, so you run HiSat taking in the raw RNA-seq reads, and if you have a reference available, you can align it against the reference annotation. And then once HiSat produces read alignment files, which StringTie uses to do transcript assembly, and then you can merge it using the StringTie merge tool, which basically consolidates all the annotation into a single file. And you can also use an accessory script like GFS compare to compare your assembled transcripts to the reference annotation ones. And then you can again run string tie but this time you can use the merged annotation from string tie merge, and along with the -E and B option, which basically will create the read coverage tables for each genomic feature. And that goes into BallGown, which does the differential expression and offers you visualization aspects. So overall, in each of these steps, wherever possible, it is recommended, like if you have a reference annotation file, so please use it. It improves the accuracy in terms of it, like its prediction. And just to give a brief intro of what each of these tools does. So HiSat, as I said, does the alignment. It has a distinct advantage where the method, the data structure it builds, where it builds both a global FM index and several small FM indexes, which makes the splice alignments much faster compared to what TopHat used to do. Similarly, string tie, which assembles the transcripts from reads, it also differs from cufflinks in the way it uses the network algorithm. That is, it simply computes the abundance and also determines the exon intron structure at the same time. And this also improves the speed compared to the cufflinks. And BallGown, it offers more flexibility, that is in terms of it provides differential expression analysis at different genomic features like gene, transcript, and exon level. And the default test it uses is the parametric F-test. It offers both multi-group and time series comparisons to be done. So it is more liberal compared to the CuffDiff method. So that's the overall picture of how the HiSat workflow can be run. So we'll do a demo of this one, then followed by the Callisto one. So before we do the demos, just to give you again a brief idea of what Callisto principle, the pseudo alignment means, or how Callisto works. So basically, Callisto requires a transcriptome file as an input. So what is pseudo alignment is basically it maps the compatibility of a read coming from a transcript. And how fast is it? Since it has a better data structure where it produces a fast hashing and pseudo alignment, it basically improves the runtime of almost about 500 to 1,000 times than the previous approaches. And because of the speed, it allows you to do bootstrapping, which helps you to address some of the technical and biological variation that you associate with your samples. So this figure here basically describes how the pseudo alignment principle works. So basically you can see here you have the transcripts in pink, blue, and green. So Callisto basically first creates what is called as the targeted De Bruijn graph, where each node here represents a k-mer that was built from the transcript. So it basically associates each k-mer with a transcript or a set of transcripts, and it creates like a hash table where it maps for each k-mer from which config it came from, and also the location within the config. So, and then it will take the read and it will find it will take the k-mers, it will hash the k-mers to the specific nodes in the graph where it finds compatibility, and then it traverses through this graph and sees where or which nodes have. So it basically finds an intersection of the nodes where the read has compatibility, and it will avoid the nodes where there is repetitive or redundant information. Like in this case, you can see the out of the first 3 nodes, it only picks the one because the next 2 ones basically gives the same information because they're covered around the same region. And So this kind of skipping or through the graph or nodes, that's where the speed of the Callisto algorithm is, and so that's a very clever technique on how you can traverse through this graph. So the previous approaches basically would have eventually gone through this entire graph and seen what would have taken a lot of time to give the quantification results. So, okay, so we don't want to get more into the details of that. So just to give you what that pseudo alignment principle works. So we'll do a demo of this workflow along with Sleuth, which will explore the visualization aspect of it. So both of these workflows are available on Cyverse, so there are distinct advantages that you have when you use Cyverse, considering these tools have improved performance in runtime and also the memory requirement. So Cyverse offers each of these tools and many other such tools as graphical user interface, and it also provides you can create virtual machines where you can have an entire workflow installed or documented so that could be run. Some of the analysis, if it's a really big data set, you can run on a HPC environment. So, and then you can build your own pipeline, you can store and share your code, and you can push your data to public repositories. And all of this services are provided as free. So that's another advantage, that if you have a limiting computing infrastructure, you can leverage Cyverse to do some of your address some of your data analysis needs. All right. So for today's webinar, we'll be focusing on using the Discovery Environment platform and the Atmosphere cloud computing platform. So we'll basically use Atmosphere for doing visualization aspect. Okay, just to give you an idea of that, so I'm just basically giving you the results of the test data that we are going to demo, just to give you an idea of how these tools performed on the same data set. So just running those tools on the test data set that we'll be working on, just you can see the performance in terms of the runtime. So say for the same data set, Top Hat and Cufflinks took about 15 hours or so, and HiSat took about almost close to 2 hours. Callisto, it took about almost 50 minutes. So you can see the difference, marked difference in terms of the performance, in terms of the runtime. And also just looking at the aligner too, the same improvement in speed can be noticed. And in just terms of the aligners, if you see even the alignment rate, that is, if you see if the reads are concordantly aligned to the reference, you can see HiSat and Callisto have a better alignment rate compared to Top Hat. So I'll discuss this result in the demo that we'll be doing now. So for the demo, will be so under the chat option, I have already posted the links for the so this is the Cyverse wiki, where we have all the contents or the documentation for both the workflows. So I've pasted both the links on the chat window, so you can go through the steps in detail. So right now I'll just specifically demo 1 or 2 apps in the interest of time, because this running through this entire workflow might take more than 1 hour. So I'll not go through each step, but just go through this wiki and give you a little idea of how things work with this workflow. Okay, so first thing, as in the HiSat workflow, is you do align the reads to the reference. You assemble the transcripts using StringTie, and then you merge them. Then you create the ball gown input files, and then you do the comparison using the ball gown application. And then for further visualization, you use Atmosphere. So for our For our test data today, so I've basically extracted the data from a paper that was, it's a recent paper study that did a drought stress tolerance in 2 sorghum genotypes under drought stress and well-watered condition. So I basically picked up a subset of that data, and here the WW stands for well-watered and the DS is the drought stress condition. And each of these conditions have 3 replicates. And so we just want to see here the transcript abundance between the 2 conditions using this particular workflow. So all of the data that we have is staged on the community data folder on the discovery environment, which I'll be showing to you in a minute. So if you just go through this path, you'll see all the input data along with some of the analyzed data is available under this directory. So what I'll do here is I'll first show you, give a little intro to the discovery environment. So if you go on the Cyverse homepage, you can see the different services that are available for doing data analysis. So today we'll be using the discovery environment. So it gives you the little bit of features what the offers, and then click on the discovery environment. You'll probably have to log in. I've already logged in before, so just use your user credentials to log in. So on the discovery environment, you basically see on the home page the 3 tabs, which is the data tab, where it provides you, it connects you to your data store where you host your data that you need for your analysis, and then the applications. So under applications, that's where our tools are located. So, so before that, I'll just give you an intro of where the data is stored. So if you click on data, go to community data. So that's where all of the test data for some of the tools are located. So under test data, go to iPlant Collaborative. From there, go to example data and find the HiSat string type Volcom directory. So this is where most of the data sets are available for this particular workflow. You have the read files, you have the reference files, and then these folders here are specifically outputs from each of these steps. So it's already pre-computed for you. That makes it easier to demo this workflow instead of waiting for the analysis to finish. So that's where we'll be using our data from for this workflow. And under applications, so you can see here it's categorized all of the tools. So you can choose based on the topic or the operation. But the easiest method is to search the particular application from the apps. I like to switch the view to a disk view. It makes it much easier. Okay, so I want to first find my HiSat tool. So I just type HiSat on the search box. As you can see, there are many versions of the same tool. So this is something we update regularly. We'll have different versions or different parameters associated with the same tool. So for today's workflow, we'll use the HiSat index align. If you just click on the little blue icon here, you'll see the information relating to the tool, the user manual, and the person integrated the tool. So it gives you a brief idea of what the tool does. And then you can always click on that particular name of the tool. So it will open up the graphical interface for that particular application. So all of the apps follow a particular template wherein you have initially, you can give an analysis name. So we'll name this as demo. Then you have a default folder under your data store, under your home directory, where where the outputs go from this analysis. You have the information on the tool, and then comes the inputs for the application. So here, the ones with red asterisk are the ones that are marked that are required for the tool. And if you want to get more information on the particular option, you can just hover over the blue icon. It gives you in detail of what kind of input it requires. So for the HiSAT, initially, so we need a reference genome. So we'll click on Browse. It will take me to the HiSAT directory where I have my reference genome file. So I'll use that as my first input, and then I have two more files that I need to put. That's the FASTQ read1 files and the FASTQ read2 files. So since we are using a paired end library, there will be two FASTQ files for it. So first thing we'll do is we'll load all the read1 files for the samples. So you can see here, you can load multiple files by selecting the checkboxes at the left-hand side, and so that takes care of the read1 files for those samples. And then you similarly can do for read2 files. And then you want to choose the fragment library. So this fragment library type. So this is a stranded library. It's a forward reverse direction. And then you can choose the file type. So here we have a paired end library. And then under advanced options, so you can either choose to keep it default, or if you want to fine tune some of the parameters, you can modify them. The only thing that I'm going to change here is to include alignments to make it more specific for string type to do the assembly, and rest of them I'm pretty much keep it the same. And then just click on launch analysis. So once it starts the analysis, you'll get a notification on this little bell icon on the right hand side. And to monitor the progress of your run, you can click on the analysis tab, where you basically get so you can see here, you can do a refresh. I had a bunch of them running before. So you can see the one here that's running right now that we launched. You can click on this to get the status of the application that is running, or you can just click on this. If something fails, it will give you further directions on how to approach that. And just to visualize the result, to know where the outputs are, just click on this when it's finished. It will take you to the default output directory where the results are. So that's how you run a particular application in the discovery environment. So what I'm going to do now is to walk over some of the results. So from the HiSAT workflow, the HiSAT alignment, what you get is basically a BAM file, which is a binary file. It basically has the information for each sample of its read alignment, and it also does an indexing of that BAM file. So the first thing when you get a BAM file is you probably want to see how many of the reads have been aligned, or there is some issue with the alignment, what is the alignment score and stuff. So there are a lot of third-party application tools, so you can visualize it in a genome browser. So you can, from Cyverse, you can directly create like a link that can go to any of these publicly available browsers. So you can use that. I use one of the third-party tools, that is the BAM viewer from the UStar Center of Genetic Study. So this offers a very neat way of visualizing the So I just pasted that link from the discovery environment, and what it offers is a little snapshot of how your data looks in terms of read coverage. So it subsamples the data from your BAM file, and you can see information like how many of the reads are mapped and the forward and reverse span, also the read distribution of them. It takes about a minute to load the information. But yeah, so you can see here, see how many of them are properly paired, what's the fragment length or mapping quality, or the base quality distribution. So that's a neat little way of doing a quality check on the output files. So for the rest of these steps, what I'll do, so we finished with the um, alignment of the reads to the reference. The next step we'll do, I'll just walk through the wiki, and I'll probably run one more analysis, and then I'll conclude on this particular workflow. So the next step is, once you have the BAM file, you do the assembly of the transcript with string-tie. So again, you follow the same method where you search for that particular app. So here we are looking for string-tie 1.33, which basically takes in the BAM files as an input. So again, the inputs for that are staged, so we can directly go from here, and then it will ask for the input BAM files. So again, select all the BAM files together. So it will output an assembly for each sample. And then you, as I said earlier, that wherever possible, provide an annotation file. So here we provide the reference annotation. And some of these apps come with inbuilt annotations as a genome service that is available on Sybase. You can either use that option or upload your own reference. What you get is string-tie output is basically the GTF files, that is the assembly files. So here is a bit basic how the GTF file looks like. It contains the transcripts, the coordinates for the transcripts, along with the transcript, the gene and the transcript ID. And it gives you the coverage for that particular transcript, along with the normalization or expression scores like FPKM and TPM for each of the genomic features. And then you also have the gene abundance file, and also the reference fully covered transcripts that match to the reference. So once you have the files that are, or the assemblies for each sample, it's always a good idea to merge all of them into one consolidated annotation file. So this is also what we used to do with the Tuxedo workflow, where we used to use cuff merge to consolidate all the annotation files. So again, you have a separate app for that called string-tie merge. It takes in the results from the string-tie step above. That is basically the GTF file. So you can load all the GTF files together, and what you get out of is a merged output GTF file. And this is what you will use again in the next run of string-tie, which will create the ball gown input files. So it basically, instead of using a reference annotation, you're using this consolidated merged annotation, and you're running string-tie again, but this time you're selecting these two options. That is, these options create the files that are needed for ball count to do the differential expression analysis. So the output from this particular step is basically the count files for each genomic feature for each of the samples. You'll have exon level expression measurement, also intron or the junction level, and you have the transcript level, and then you have some mapping files associated with that. So this is what is needed for ball gown to do the differential expression. So it basically, ball gown is an R package to do the differential expression. So what ball gown needs essentially is these CTAT files from here, And the other thing it needs is a design matrix. So the design matrix here basically specifies what samples you have. It can be very flexible. You can have any information relating to your experiment written. But here we are using the basic three information, that is the sample ID, the condition, and the replicates that we're going to use. And what we are trying to do here is to do a pairwise comparison for differential expression under the drought stress and the well-watered condition. So again, the inputs are staged here. So I just run this ball gown application for you, just as another method of doing. So again, I go ahead and search for ball gown. And again, you can name it as you want. So here here it will ask first for the design matrix file, which is again here. And then here the experimental covariate of interest is basically the name of the column that is here under which you want to do the differential expression. So make sure the name here matches what you put here. And then basically the directory of the CTAB file, which will be under the second run of string tag, which was done using the merged annotation. So you just select this ball gown input file and just click on launch analysis. So this step should be pretty quick. And then if you see, once it finishes, it will give you these files, which are basically, you'll get a box plot of the FPKM distribution. Then you get result files both on the gene and the transcript level with just the raw differential expression file. Then there is some filtering applied based on the p-value, and also filtering based on those which have low expression values. So you can, this is just the raw output from it. So the next step from here, you want to get more meaningful information from it. So that's where you can take this output and you can run again ball gown in a more R-like environment. So for that, we use the Atmosphere Cloud Computing Service. So I'm going to explain this particular aspect of how you can launch an Atmosphere image to do this kind of analysis when I explain the Callisto workflow. But if you just follow this particular workflow stepwise, you'll probably launch an RStudio, and then you can follow these steps. It's an R markdown which explains basically each step on how you can further explore your analysis to get more meaningful information from that. So I've just documented the steps that could be relating to this data. So once you actually start doing analysis, you'll explore more details. It's just copy pasting these commands onto the R console and you'll get the results from that. Okay, so I'll quickly move on to the next workflow, that is the Callisto and Sleuth. Again, we'll be using the same data set, so I don't need to explain that one again. So Callisto pipeline, it works in two steps. That is, it does the indexing so the input is basically a transcriptome file, and then from that it does the quantification for each sample, and then you'll use the sleuth for doing gene or transcript level differential expression. So again, the method is the same, where you search for the application. So here the data for that is stored under Callisto Sleuth folder under the community data, and we have the cDNA as the input for this, followed by the reads. Again, the reads here were both FASTQ files in zip format. You can also use like unzipped files, which is fine. And then you can set the number of bootstraps that you want to use. So bootstrapping helps in removing some of the uncertain estimates. So typically for a eukaryotic genome, about 30 to 60 bootstraps is good enough to to do the analysis. And again, click on launch analysis. The output that you get here is the abundance files. So basically it gives you a transcript level abundance, and you can use this. So this is what you use with Sleuth. And the H5 files basically has information again on the bootstrapping. So this is, you can't view the HDF5 files. It's basically the TSV file which gives you the raw counts of for each transcript. So the next one is you take the output from Callisto and you run Sleuth. So here I'll show you how you can run Sleuth on the Atmosphere cloud computing platform. So what Sleuth offers, it both transcript and G-level comparison analysis. It uses the bootstraps that were done through Callisto for correcting some technical variation in experiments. So there's a nice paper if you want to review some of the statistical methods for that. All right. So first thing is to connect to Sleuth, to connect to Atmosphere. Just go back to the Cyverse homepage, where you can click on Atmosphere, launch Atmosphere. So it will open up the so already logged in. So here you can see you have click on launch instances, where you have the different images, and you can search any image relating to your analysis. There are images specific for RNA-seq that you can launch, or you can basically go ahead and for this particular exercise, we'll be using the Ubuntu base image. So once you click on that, you'll get a dialog page where you can assign the resources that you want to run instance for. You can set the size of your instance to whatever requirement. If you have a big data, try to have at least a medium or medium to kind of an instance size. Just give a name for your instance. And once you click on launch instance, you'll see on the project page the information or the status of the instance if it's active. So there are many ways you can do the run this particular instance. You can just copy this IP address of the instance, and if you are working on a Mac or a terminal on a PC, you can just do an SSH with your username with the IP address from above, and it will ask you for a username and password, and it will connect you to your instance. And there it's basically as if you're running your own version of your terminal on the cloud. And the next step is to stage the data for Sleuth analysis. So you create a directory and you use icommands, which is basically the iRODS package for doing data transfer. So you can use that to connect your data store and download the files that needed for Sleuth. So here I've documented the steps. Again, it will ask you your Cyverse password. So once the data is staged, the next step is to run RStudio in Atmosphere. So these are some of the commands to launch the RStudio. And once you run this particular step, you'll get an link which you can paste it on the browser, and you'll sign in again using your Cyverse credential, and it will open up an RStudio page where you can do your analysis like what you do on a standalone RStudio tool. So again, so I have documented all of these steps here on how Sleuth works. So again, I'll not go through all of this in detail. So I'll tell you like up to, so basically Sleuth reads in all the information from the Callisto thing. The one more additional input that I've given here is the target to gene mapping, where basically I'm adding information to gene IDs to the transcripts. And then what did Sleuth, the Sleuth steps, it basically runs two statistical tests, that is the likelihood ratio test and the Wald test. And each of them offer different statistical performances. The easiest way to do or visualize the results is using the Shiny application, which Sleuth offers. So here, so this is the virtual machine that I'm running the RStudio on. So once I did the Wald test under the condition well watered, and then I can just run this step, which will open up the Shiny application relating to Sleuth. So you can see various different diagnostic plots here that is associated with Sleuth. You can check the mean variation plot, which basically gives you the abundance versus the standard deviation of the counts. And the red line, the blue dots here are the interquartile range, and the red curve is basically the Sleuth fit for the shrinkage estimation. So this basically gives you an interactive way of visualizing your results. So here under the scatter plot, you can see how your samples are correlated. You can choose from this, and it generates the output for each one of them. You can highlight specific data points, and you'll see how the variation for the data points happens here. And some of the details for the particular transcript selected are populated in a dynamic table. There are other diagnostic plots like QQ plot. So that's basically a plot of how, if there are underlying data is coming from the same model. So you have the observed quartile on the y-axis and theoretical quartile on the x-axis. The red here indicates the differentially expressed genes. You can set it for any FDR value under any conditions, so it dynamically updates those values. And there are other, so you can do a PCA plot. So that's, again, you can see how your samples are distributed. You can do it based on condition or reps. It will give you how the variation is explained on the for the samples. And there are many other such applications. You can do a volcano plot. Again, you can so the red ones here are the differentially expressed ones. So you can see this is a plot of a natural log versus the beta value. So beta value in Sleuth terms is basically a fold change value. You can select those, and it auto-populates them. Then there are other ones. You can do a gene view. So you can basically, let's say, maybe I'll just get an ID, a gene ID from the table. So you can just copy this gene ID if you want to see the abundances across the different conditions that we want to see. Sorry, maybe I should copy one which has some meaningful information. Okay, there you go. Gene view. All right, so that basically gives you an idea of how the abundance for that particular gene is across the two conditions that we have. So you can see there is a little bit of variation with rep1 and rep2 here. So Sleuth basically offers you a nice interactive way of summarizing your results and doing analysis. So it's pretty neat. So this is just, so you can leverage the cloud atmosphere to run Sleuth, or also you can use the RStudio to do that. So I'll just stop here now. So we're almost closing to the runtime. So again, I'll just show you the results. We have, so you can see the runtime again for the workflows, the alignment rate, and so so just to summarize, again, so the new and pseudo alignment RNA-seq protocols offer a much faster way of doing analysis for RNA-seq data, reduces some of the bottlenecks, and the differential expression packages, especially Bolgon and Sleuth, offer good interactive visualization. There are other tools like STAR, Salmon, and DSeq2, which also are good alternatives. These are also available on the Sciverse environment. And so again, the bottom line or the take-home message is like the choice of your pipeline that you do does affect to some degree on your downstream results. So it just stick to the standards, only deviate when it's necessary. Again, think about how the field of RNA-seq or the technology, the sequencing technology is changing. So there is always some newer tool that comes which performs better. So it's always better to take advantage of such tools and leverage it on platforms that offer you easy access to such resources. And then RNA-seq again is a guiding tool. So you want to experiment. I mean, you want to validate some of your results with some experimental methods. And Sciverse again is will be adding a lot of some of RNA-seq tools, and will probably have some more visualization aspect, like what I showed on Sleuth. So if there is a need, or so we can do one more such kind of webinar where we focus mainly on the visualization aspect. So overall, so that was the brief overview of every workflow there. And if you have there are some resources. If you have some questions, you can go on the Sciverse Ask Forum, where we have details on the some of the questions asked by the users on the workflows or any of the tools. And there are also a lot of community resources like BioStar, Seq, Answers, RNA-seq blog, which are very informative on such analysis. And yeah, so thank you for listening. And I think we have time, 5-10 minutes to go over the questions. So before I go take the questions, just to remind people, like we have the next Sciverse Focus Forum on the data store, the Sciverse data store. The details will be posted on the Sciverse homepage. So this will basically show you how you can use the data store for your data analysis. And finally, we would like to thank the funding agency, National Science Foundation, and our institutes that keep Sciverse running. All right, so thanks for listening, and I think I'll go ahead and look for questions if there are any.
Thank you, Kapil. There are 2 questions posted in the chat. Can you see those?
Yes. How do you get a merged GTF to CTAB files for Bolgon? Okay, so I think if I go back to the HiSat workflow, yeah, so once you do the string time merge, so you get this where is that? Yeah, the merged output file. And so you'll have to run string time again, as I said, but this time you'll be running string time with these 2 options, that is -B and E, which is highlighted here with the 2 tick boxes. And here under reference annotation, you use this merge out GTF file. So that basically is a consolidated annotation file rather than just giving the reference one, because you already did an assembly with string tag for all the samples. So you kind of merge that with your reference annotation file. And then after you run this string tag step, you will get the CTAB files under this directory of Bolgon. So that's basically what you use for your next analysis. Okay, the next one, can we visualize Callisto outputs on Genome browser? If not, how do we visualize these? So I think the best and the easiest way to, if I understand, the latest version of Callisto does offer a BAM output. I haven't tried it yet, but that's something like what I showed with HiSat. You can just copy the URL and use either this kind of tool, or you can just go on a Genome browser that you can probably use to visualize. So yeah, so you can. I think Callisto has a new option where it gives the pseudo alignment in terms of a BAM file. So that's something. Either I can have a newer app for Callisto that does offer that feature. All right. I think, yeah, there's no more questions from there. And yeah, so all the materials will be available on the wiki. So just follow through all of the details. If you have any questions or comments, you can either post directly on the wiki link at the bottom. So you can just post comments here. And yeah, so and then if you have any also questions, you can directly contact me as well.
Kapil, there's one more question. What is the name of the tool you used for visualization in the tutorial?
So for the Callisto one, it was the Sleuth. So it's the R package, which offers both visualization and features. So I basically use the R Studio as an environment to run Sleuth, which basically, if you run each of these steps, it offers under the R environment the visualization libraries to do the plotting of the data sets. And then it also, in terms of ball gown, you also have the same thing. It's R Studio. That's the tool that's used on the Atmosphere.
Thank you very much, Kapil, and thank you everyone for attending. We'll follow up with an email.
Sorry, I think the question was.
Oh,
yeah. So it's the tool that I used here was the BAM IO Bio. So I can post the link over here just in case. Sorry, Tina.
no, thank you for catching that.
Okay. All right.
Okay, we will be following up with an email that has a link to the wiki page where all these materials will be posted. And we just appreciate you attending. Thank you, everybody. And please join us for the Data Store webinar in September. Bye now.
Thank you. Bye.